my title is a little different. Hopefully, it's not confusing you. The title that I have today is Potential Factors for Variable Responses to Feeding Amino Acid with an emphasis on lysine. Dr. Hannigan mentioned that if you provide amino acid to lactating cows, I agree with him that if amino acids are delivered well, then cows will have a response. But if you look at the individual studies, that's not always the case. So a little bit of an introduction before I go into that topic. So balancing a diet for amino acid. Now we have models available that we can use to formulate a diet to provide adequate amount of amino acid based on the model. We can use those models to formulate a diet. And the goal of the balancing a diet for amino acid is probably to increase the efficiency of protein synthesis. So if that is the case, then we can avoid excessive supply of nitrogen through the diet, and also we can reduce nitrogen excretion in manure, which means we can provide the same amount of nitrogen through the diet, then we can increase the production if we provide more balanced amino acid, or we can reduce the dietary protein with the better amino acid profile, then we can maintain the production with less amount of protein supply through the diet. So the goal, ultimately, we are hoping to see to increase the income over feed cost and also decrease the environmental impact. So the updated model, those are available. We use those to formulate the diet for amino acid, but still it identifies often, depending on which feed ingredient you use. The methionine and lysine are often a limiting amino acid. And if you use the NISAM, you often probably see histidine is also another limiting amino acid. So historically, before the new models, we used the MP-based system to formulate the diet for protein. And it often estimated the methionine and lysine are co-limiting amino acid. After that, we conducted lots of studies, and publications are available about feeding rumen-protected methionine and rumen-protected lysine. And studies with the rumen-protected histidine are much more recent, relatively recent. But we have quite a bit of studies. And with this accumulated data, scientists try to look at their effect through the meta-analysis. So this is a table that includes the meta-analysis that you can find in the literature. I think those are the most of them. And most of meta-analysis were done with methionine, because the methionine was the studies that was conducted more than any other amino acid. And then if you look at those studies, the results are pretty good. So which means this is very good for us. If you provide limiting amino acid, cows will respond based on those meta-analysis. But if you look at the lysine here, so one published in 2010, this paper is just accepted in Journal of Dairy Science. It's in press. This first meta-analysis, actually the effect of lysine was not positive. So there was no response to feeding additional supply of lysine. This study included lysine infusion and feeding rumen-protected lysine. The last one, it included only rumen-protected lysine studies, and it showed that meta-analysis found that they increased the milk yield and increase the milk protein yield. So let's look at those two studies a little bit more. So one published in 2010, it included the 7 studies with about 24 treatment. It included the studies with the lysine infusion and also feeding rumen-protected lysine. The last one The one that just accepted from Wisconsin. Third, it included 13 experiments with 40 treatment, and it included only studies with feeding rumen-protected lysine. I still think the number of studies are quite low. For meta-analysis, I think more studies are needed to have a better evaluation of the lysine. But that's not what I'm trying to point out. What I'm trying to point out is the difference in the result between this meta-analysis and this meta-analysis. There are probably a lot of reasons about why the results are different. The methodology is different, number of studies are different. The studies selected to be used for the meta-analysis are different as well. But I'm not going to talk about all those. But what I'm trying to talk about is why they are different. There might be other reasons or factors that cause the variable responses to feeding rumen-protected lysine. If you look at the individual, in this meta-analysis, if you look at the individual studies, most of them had no response to feeding rumen-protected lysine in individual studies. So why is that? So it's not just rumen-protected lysine. If you look at the papers about rumen-protected methionine or rumen-protected histidine, there are good responses, but also you can find studies with no responses to feeding rumen-protected amino acid. So there are many studies, but I was not able to include all the studies in this table, but I brought only transition cow trials with feeding rumen-protected amino acid. I was able to find those transition cow trials in the Journal of Dairy Science and some other journals. But what I'm trying to point out is, other than those first 3 experiments, the rest of the experiment didn't see any response to feeding rumen-protected methionine or the combination between rumen-protected methionine and lysine. The first 3 experiments is very interesting. They observed the feeding rumen-protected methionine increased milk protein yield and milk protein yield, milk yield and milk protein yield. However, they also found increased dry matter intake. I don't know how methionine increased dry matter intake in this experiment, but in those studies, there was no difference in feed efficiency. So I assume increased dry matter intake was the main driver for increased milk yield and milk protein yield. So what I want to point out in this table is still there are variable responses to feeding rumen-protected amino acid. So things to think about in terms of feeding rumen-protected amino acid. So we have models available that we can use to formulate the diet to provide good profile of amino acid, good amount of amino acid to the lactating cows. If we use those models, we know we can identify easily which amino acids are limiting. And we can use different feed ingredient or different rumen-protected amino acid to provide adequate supply of certain amino acid. However, if we look at the individual studies, as I showed, still we have a minimal effect or no responses or good responses. Responses are quite variable between studies. So especially from rumen-protected lysine, based on my opinion. So supplementation of rumen-protected amino acid is pretty common in commercial dairies, and rumen-protected amino acid is not cheap. We cannot just dump a lot of rumen-protected amino acid in the diet to make cows respond. We have to think about economic return as well. So my opinion is for the next amino acid research for lysine and also other amino acids as well, we need to identify factors that can cause the variable responses to feeding rumen-protected amino acid. if we can find that the factors, we should spend time to fix the problems and increase the response and make the response more consistent. So if you ask me what those factors are, I don't know. I don't have any answers. However, based on the information in the literature and some of our previous studies, I'm trying to come up with, share with you potential factors that might be the factor that cause the variable responses to feeding rumen-protected amino acid. So first, the factor that I want to talk about is the flexible amino acid utilization by tissues. So lysine is one of the group 2 amino acid. Group 2 amino acid include the lysine and then branched-chain amino acid. I'm going to just talk about lysine here. Group 2 amino acid has a characteristic that after absorption, lysine is not extracted or utilized by the liver. However, once it's taken up by the mammary gland, the amount of uptake is always greater than the lysine output in milk. So the ratio of the output to the uptake to output is greater than 1. In this experiment, so the group 1 amino acid that include methionine and histidine, that's quite opposite. They are a part of those extracted in the liver after absorption. And then the uptake by the mammary gland and output in the milk is almost the same, so the ratio is 1. But lysine here, as a group 2 amino acid, are taken up more than the output in milk, which means maybe the question that we can ask for that is, where does the lysine go in the mammary gland? It was taken by the mammary gland, but the amount of lysine secreted in milk is always less than uptake. So where did the lysine go? So this is a study published in 2009 from Dr. Lapierre's group. In this experiment, they infused the complete amino acid mixture with and without lysine into the ovomasum. And at the same time, they infused M15 lysine into the blood. And then they collected blood sample and milk sample to look at the M15 enrichment in amino acid of blood and milk casein. So any values here is pretty busy table, but any values with highlighted in red indicate significant 15 nitrogen enrichment. So again, they infused the 15 nitrogen lysine into the blood. So any values highlighted in red indicate a significant 15 nitrogen enrichment, which means somehow lysine infused into the blood was metabolized and then utilized to produce other amino acid. So this is the good evidence that body tissue and mammary gland, especially mammary gland, has a large flexibility, at least to utilize lysine to produce a milk protein, to produce other amino acid, and utilize it for protein synthesis. We conducted a similar experiment. We infused the M15 lysine into the ovomasum. We collected the plasma, blood, and then milk protein, and look at the 15 nitrogen distribution across the amino acid in there. And any values highlighted in red indicate significant 15 nitrogen enrichment. So basically, most of amino acid were labeled with 15 nitrogen. It looks like it's not just the lysine. Based on the literature, the branched-chain amino acid perform like lysine. So branched-chain amino acid also utilized to produce other amino acid, and those amino acid is likely used for protein synthesis. So lysine oxidation and then transamination, those are the process to produce other amino acid I don't know if this is the required process for the protein synthesis, or just is occurring because lysine supply is excessive. I don't know. But we observed this process in the mammary gland when lysine supply was deficient. Okay? If this is the required process, we believe we need more study to understand this amino acid metabolism, to better understand how the requirement of those specific amino acids. And leucine, isoleucine, from Dr. Hannigan's group lab, it was found that those are, as I mentioned, those are utilized to produce other amino acid, and also those amino acid has a different role of stimulating protein synthesis. So it's not, I agree with the previous speakers, it's not just a protein building block. We have to understand other important roles of amino acid to understand the requirement of amino acid in lactating dairy cows. Okay, second potential factor that I want to share with you is a different requirement of amino acid between lactation stages. This is a table that I showed how variable the response is and when rumen-protected amino acids were fed in the studies, in the literature. And this is only transition cow trials. And why a fresh cow? I believe fresh cows respond, the response in fresh cows is more variable compared with early or mid-lactation cows. So probably it's obvious because they have a low intake, there are a lot of cow-to-cow variation, and there are a lot of mobilization of nutrient during the transition fresh period. So that's probably why more variable responses are occurring in fresh cows when rumen-protected amino acids are fed. But I think there is something else. There is something else. That's what I'm trying to share with you. Is there a priority for amino acid utilization over milk protein synthesis? We think a nutrient absorbed in the gut is utilized as a priority for milk yield or milk protein synthesis. But in fresh period, maybe there is something else. We assume that fresh period is a very stressful period. Fresh cows may be under inflammation state to some degree. So if this is true, there should be an immune system activated and maintained to some degree in this period. There is a great work for the activation of immune system, maintaining the immune activity, they need the nutrient. There is a great work from Iowa State University. They tried to come up with energy requirement to maintain for the immune system in lactating dairy cows in the short term. This is a study. I'm not going to talk about all the details, but what they found is under the inflammation state, maybe nutrients absorbed in the gut are utilized for immune system as a priority over the milk protein synthesis or milk yield. So maybe amino acid might be the same. I don't know yet, but probably it could be similar. And we found a little bit of evidence for that. This is the experiment that we conducted before. It was a 4x4 Latin scale design. We infused M15 lysine into the abomasum. This is from one specific cow, one particular cow. So in period 2, somehow that cow dropped the intake and milk yield. The vet checked the cow, but we didn't find any disease or anything. But she dropped the intake and milk yield. We assumed she was sick during that time. But period 1, 3, and 4, she was okay. The intake and milk yield were well maintained. As I said, we infused M15 lysine into the abomasum. So if we did the total collection of urine, milk, and feces, we calculated the 15 nitrogen balance. This is the result of 15 nitrogen excretion in urine. This is the 15 nitrogen retained in the body. So the blue line represents the cow that was sick in period 2, and orange line is the same cow, but when she was not sick in period 3. So as you can, if you look at the excretion of 15 nitrogen that we infused, so when the cow was sick, 15 nitrogen excretion in urine was significantly, considerably increased. Again, I don't have any statistic for this data. But for the calculated retained 15 nitrogen was decreased significantly, considerably, by 15% unit. We have to look at do the more designed experiment to confirm this result. But I think this is the good evidence that if that cow was really sick, and if there was inflammation going on during that time, and if that for the immune system required nutrient to some degree, then probably the infused lysine was utilized as amino acid or as energy. But in this case, because that nitrogen was, and 15 nitrogen was excreted considerably in urine, my interpretation is probably it was utilized as energy, and then nitrogen was just excreted in urine. So this is probably good evidence as well that there is something that we have to understand for fresh cows. If fresh cows are under a certain inflammation, and if that is require quite a bit of nutrient, we have to understand that to improve our prediction of amino acid requirement. The third one that I want to share salt factor that I want to share with you is the varying prediction result between models. So I'm going to skip this. So what I want to share with you is probably all of us rely on those two models, NICEM and CNCPS. We either use NICEM or CNCPS, and we rely on those models to formulate the diet. And we formulate the 3 different diets. One is the MP adequate diet, MP slightly deficient diet, and MP deficient diet. And then we estimated the supply and requirement between NICEM and by the NICEM and CNCPS and compare the result. And if you look at the methionine balance, it's pretty much the same, which is very good. Whatever you use, NICEM or the CNCPS, the amount of rumen-protected methionine that you are going to include in your diet will be the same. However, lysine was slightly different. For high-protein diet, the lysine balance was about the same. It was not deficient. But as the protein concentration decreased in the diet, the deficiency was different. So depending on which model you used, the amount of rumen-protected lysine that you are going to include in the diet will be different. That amount, depending on which one is more accurate between those two models, even though you included the rumen-protected lysine in the diet, the metabolizer lysine supply might be still deficient, or it could be excessive. What I want to point out, one more thing, is histidine. So the large difference that we found between those two models was the histidine. The deficiency was found by NICEM for most of diet, but deficiency was not found by the CNCPS for most of the diet. So there was the more, but I'm not, histidine is not my topic, so I'm going to skip it. Another thing that I want to talk about is the meta-analysis that I introduced that was just accepted in the Journal of Dairy Science. They look at many different things about rumen-protected lysine. One of them is lysine supply as a proportion of metabolizable protein. In this meta-analysis, what they found is milk yield increased linearly from 6.5 to 8.5% of lysine as a proportion of metabolizable protein. So if you remember NRC 2001, the recommendation was 7.2. And if you use the NISM, if you provide adequate protein, metabolizable protein, and lysine, the proportion is about 7.3, 4, 5, or something like that. It's about the same as NRC, very close. But if these results are the true, so this meta-analysis found that the milk yield increased until 8.5%. If this is true, then lysine requirement might be greater than predicted by the current models. So I think a little bit of things that we can do in the next probably 10 years, that we can do more studies, understand better, and about the lysine in addition to other amino acids to improve the prediction of the amino acid requirement. The last thing that I want to mention is the bioavailability of rumen-protected amino acid. Dr. Hannigan talked about those, and I totally agree with him. It's very important that we have to know the right the bioavailability for the product. Feeding rumen-protected amino acid with incorrect bioavailability may lead to a deficient or excessive supply of a certain amino acid, which means you included rumen-protected amino acid, but if that amino acid is still deficient, then there is no response from the cows. If you have excessive supply because of the wrong bioavailability, then your economic return is very small. So this is a good example. I like this study from Penn State, from Dr. Hrisup's group. I'm going to just point out rumen-protected lysine here. The group tested many different rumen-protected amino acids, tested the 3 different rumen-protected lysine. The group developed a method to measure the bioavailability, and those measured bioavailability was compared with the bioavailability provided from the manufacturers. So here, dark bar represent the measured bioavailability, and gray bar represent bioavailability from the manufacturers. So as you can see, this one is pretty close, but all the lysine products was overestimated based on this study. So if this is true, then even though if you use this information from the manufacturer, you put the rumen-protected amino acid in the diet, but most likely that amino acids are still deficient, right? So then your response, no response or variable response makes sense if this is the case. So this is the last fact that I wanted to share with you. And in summary, as I mentioned, we have models. We use those models to formulate the diet to provide good profile of amino acid and amount of amino acid. And feeding rumen-protected amino acid is pretty common, but we have to think about the better response and more consistent response according to the studies in the literature. Responses to rumen-protected lysine are more likely variable compared to other rumen-protected amino acid, but the result from the recent meta-analysis is pretty promising, and I hope that that is true, that the results are true, and there is other things that we should do in the future. As I mentioned, we have to find the factors and try to fix the problem and try to have a better response and have a more consistent responses to feeding rumen-protected lysine. I mentioned about several factors. I'm not saying those are the factors. Those are the potential factors based on my experiment and based on the information in the literature. We have to understand better about the role of amino acid other than protein building block, so we can in the future can include those information in the model to improve the prediction of the amino acid requirement. And also the stage of lactation is very important. They are all different, I think, amino acid requirement with many factors. I think one of them is immune system. And determining accurate bioavailability is also a key factor that we can make the response more consistent. So those are the factors that I want to share with you in this presentation. This is all I have.